Plants Part 1, the second part of it, sampling and propagating plants. So if we begin by looking at keys, these are used to identify organisms, and there are two types. There's branching keys and there's what's called paired statement keys. If we have a look at branching keys first of all, let's have a look at these two cows. Cow number 1, for example, is the body more than one colour? Yes, it is. Does the animal have a black or white face? It has a black face. So cow number 1 is a Russian black-pied. For cow number 2, is the body more than one colour? No, it is not. Does the body have long or short hair? It has long hair. So cow number 2 is a Highland cow. Now, paired statement keys, they follow exactly the same process as branching keys, except the questions are written out in sentences rather than presented as a diagram. So let's use an example. Let's use animal J as an example. Does the body have legs? Yes, it does. Go to number 6. Jointed legs present? Yes, they are. Go to number 7. Grub-like insect or an adult insect? So it's an adult insect at 9. Thin waist between the thorax and abdomen? No, it is not. No thin waist between the thorax and abdomen. Go to number 10. Spring attached to the abdomen? Nope. No spring attached to the abdomen. It is a beetle. Now let's have a look at factors which affect the survival of organisms. We've got biotic and abiotic factors. Biotic factors, these are caused by other living things which can affect the survival of living things, such as competition, predation, grazing and disease. Abiotic factors, on the other hand, are factors which affect survival of organisms, but they're caused by non-living features of the environment, such as light intensity, temperature, pH and moisture levels. If we have a look at a couple of examples of measuring abiotic factors, whenever you're using a light intensity meter, you need to make sure that if you're measuring sunlight, you need to record it at the same time of the day, if it's over several days, and of course make sure you don't stand in the way of the light source so you cast a shadow onto it. Moisture meters, you need to make sure that you push the probe the same depth into the ground each time, and make sure you give it a good wipe after each use so you don't affect subsequent readings. Now, for sampling plants in the environment, one of the favourite methods is using a quadrat, and we use them to measure either the abundance or the size of population of plants in a certain area. Now, if you imagine this is a field full of daisies, for example, first of all you need to measure the actual area of that field, and that will give you the area of the field in meters squared. Next, you will throw your quadrats randomly and as many times as possible in that field. And then if you just take your average number of daisies in 1 meter squared quadrat and multiply that by the whole area of the field, that will give you a population estimate of daisies in that field. Now, in order to minimize errors when using a quadrat, you have to make sure you throw it randomly and throw it as many times as possible. Now, another way of using a quadrat is by doing a transect survey, and that's looking at the changes in organisms from one habitat to another. Now, instead of throwing the quadrat randomly, you place it at regular intervals and count the number of organisms inside each quadrat as you move from one area to another. Now, another thing to use quadrats for is measuring abundance. Now, instead of making an actual population estimate, what you're doing is just comparing the populations of different animals, different organisms, plants, by looking at the number of squares in the quadrat in which they appear. Now, for example, if we were going to look at the abundance of daisies, we would count the number of small squares that the daisies appear in. So there's 1, 2, 3, 4 out of 25 squares within the quadrat. Okay, now if we were going to look at the abundance of dandelions, for example, the dandelion flowers at least, appear only in 1 square out of 25. And that's how you estimate the abundance of plants in a quadrat. Now, if we move on to look at plant reproduction, if we consider first of all sexual reproduction in plants, that involves gametes. They are the sex cells, which in a plant's case will be pollen and egg. 2 parents are involved in this kind of reproduction, and the offspring will show lots of genetic variation. However, if we consider asexual reproduction in plants, there are no gametes involved. There is only a single parent, and all the offspring are genetically identical to that parent. In other words, they are clones. Now, if we have a look at the different ways plants can asexually reproduce, which is called vegetative propagation, there are 5 different methods. There are bulbs, like onions. There are runners, such as that which you find in a strawberry plant. There are offsets, like that which you find in this case, a water hyacinth, or in class, the mother-in-law's tongue. There are tubers, like a potato. And there are plantlets, like that which you find in a Mexican hat plant with the tiny little plantlets at the edge of the leaves. Now, if we have a look at artificial propagation, this is the way that we, humans, produce clones of plants that the plant can't do naturally. There are cuttings, such as leaf cuttings or stem cuttings. There's layering, where you can peg the stem of a plant down so it starts to grow roots, and then you can cut it off and it'll grow independently. There's grafting, where you can take the stem of one plant and physically insert it into the rootstock of another, and once the bond is sealed between the 2 parts of a different plant, they can grow. And there's tissue culture, where you can produce copies of plants inside the lab. If we have a look at the stages in tissue culture, first of all you take your samples from the parent plant and you grow them in a nutrient solution called agar, where the cells reproduce. You then take the tiny little clumps of cells and they develop into tiny plants, and then finally you just take those tiny little plants and grow them naturally in compost, and they grow normally. Now, the advantages of using tissue culture: we have what's called the guarantee of uniformity, which is basically meaning they're all going to be exactly the same. So if you have one really good type of plant that grows really well and produces lots of fruit, for example, you know that all the offspring from that plant are going to produce the same quantity of food. We can also reproduce sterile varieties. These are food plants without seeds in, such as grapes and bananas. And of course, any rare or endangered plants can be propagated in this way to help with conservation. Now, if we move on to look at germination in seeds, you already know that seeds need water, oxygen and warmth, or a suitable temperature, to germinate, and this is the wow factor. There are several techniques that you can use to try and get seeds to germinate. The first one, chitting, where you take a little knife, you can cut the seed coats so water soaks into it. You can either pre-soak them in water, or you can give the seed coat a little scrape with sandpaper to help the water seep in to help promote germination. And finally, we come to compost. Most composts that you buy contain something called loam, and that's basically good garden soil. And they contain things like peat for water retention, or a substance called perlite, which allows air to get to the roots, or sand, for example, to help water drain from that compost. You can also get loamless composts, and these have peat instead of loam, but they need regular nutrients to enable the plants to grow. Okay, thank you very much.